There are times when evolution is not actually happening, and we call this genetic equilibrium. It is also known as the Hardy-Weinberg equilibrium from the two people who discovered it and came up with the idea. So this is when there are no changes in the allele frequencies in a population over time. So if we can document that there's no changes, we can verify that there is no evolution happening to that population. So we're going to look at a list of things that if these are not happening, we know that evolution does not occur. So if we start looking at our list over here, the first thing that we have is that the population must be large. So when we have a large population, it will prevent evolution from happening. So the smaller the population, the more likely evolution is to occur. Next is there must be random mating. So the opposite of this, we would not want sexual selection to be happening. So if we're selecting something for a reason versus just mating randomly, that would be preventing evolution. Then we have no migration, no mutations, and no natural selection. These three things have in common the preventing the new sources of genetic variation. So if we look at this list of 5 things, if at least one of these conditions is not met, then the population is evolving. So if we have all of these 4 things, but we're in a very small population, then it is most likely going to be that evolution is happening to that small population. So when we're using the Hardy-Weinberg equilibrium, we know that evolution is not occurring. So we're trying to figure out if we can determine this for a population or not. So we already know that in all populations with only 2 possible alleles for a gene, we're going to use this equation. So if there's 2 alleles, one of them will be P, the other one will be Q, and together they equal 1. This equation can allow us to calculate the allele frequencies for the dominant allele, P, and the recessive allele, Q. If this frequency does equal 1, then we know it is in Hardy-Weinberg equilibrium, so we can use an additional equation to calculate the frequency of the genotypes in a population, which is going to be this equation of P squared plus 2PQ plus Q squared equals 1. So I know you're like, why am I having to do math in biology? You have seen these types of equations before, so this is simply just a factored version of P plus Q squared. So you have seen these things before. It's not going to be that difficult, I promise. So looking at this equation, let's try to determine what each of these letters mean. We already said in the last slide that our P is our dominant allele and Q represents our recessive allele. So if we pick B to represent this, we know that P could be our big B and Q is our little b. So P squared, if that's B, it is the genotypic frequency of the homozygous dominant individual. So if we remember, homozygous means homo, we have the same, and dominant is big. So that would be big B, big B. Then we have 2PQ, and this is a P and a Q, so it's the genotypic frequency of heterozygous individuals. So this would be representing our big B, little b individuals. And then lastly, we have Q squared, which is the genotypic frequency of homozygous recessive individuals. And again, homozygous means same, and recessive means little. So now we are going to have little b, little b. So we're going to look at our example 3 here and try to figure out what are the allele frequencies for this population. So we have round heads being dominant to cone heads, and we have 51% of the individuals having round heads. So if we think about this, round heads can be either having big R, or big little for round, and then the opposite, if it doesn't have a round head, a cone head, would be little r, little r. So we want to know what portion of this 51% are homozygous if the population is in Hardy-Weinberg equilibrium. So we're trying to determine what percent of them are this big R, big R. So as we said, we have 51%. That means that 0.51 of the population shows the dominant trait. So they're either going to be big, big, or big, little as their genotype. There's 100% of the population, so we can just do the math, 100 minus 51. So we know that 49% of the rest of the population then has to have a cone head. So we know that this 49% of individuals have this little r. And just to remind ourselves, we know that this little r is representing our q squared that we saw on the last slide. So then we can say that our 0.49 is our q squared. So if we're just trying to find q, we're going to want to find the square root. So for q, for us in this example, our q is going to be 0.7. And if p plus q must equal 1, we know that p then is going to equal 0.3. But we are not just trying to find p, we want to know what of this 51% are homozygous. And that is of these two up here, so we want the ones highlighted in gray. We want big R, and that is represented by our p squared. So then we want to take this number down here, our 0.3, and square that, so multiply by 0.3 again. So we will have our p squared as 0.09 or 9%. So 9% of our population is big R, big R. For example number 4, we are looking at turtles, and our red coloring is going to be dominant to yellow color. We have 241 turtles in our population, with 34 of them being yellow. And we're going to assume for this scenario that they are in Hardy-Weinberg equilibrium. So we have two different questions here. First, we want to find the allele frequencies, which is using our p plus q equals 1 equation. And then we have what percent of each genotype is in this population. That is when we have to use the slightly more complicated one. So first we want to decide what our letters are. So we have red as being dominant and yellow as being recessive. So we are going to use our dominant letter. So big R is going to be for red, and our little r is going to be for yellow. So let's first determine what percent of our turtles are in this yellow phenotype. So we know that we have 34 of them. So we are going to take 34 and divide by 241, the total number of turtles. And this gives us an answer of 0.14. And again, this is for our little r's, which is giving us the yellow phenotype. And our recessive letters, our two little letters together, is what our q squared is. So now we want to find what just q is, not q squared. So we're just going to take the square root. So we want the square root of 0.14, and that is going to equal 0.37, which is our q. So now that we have figured out q, we can use this to help in our original equation of p plus q equals 1. So we know what q is, so we are going to take 1 minus 0.37 to figure out what p is, and that is going to equal 0.63 is p. So just a reminder before we move on to part B, that allele frequency is when we are using p plus q equals 1. And allele frequency is how often these alleles are in our population. Okay, and now on part B, what percent of each genotype is in this population? So I first want to remind ourselves that we are going to be using the equation of p squared plus 2pq plus q squared equals 1. I'm also just going to jot down the information we figured out on the last slide because we will need to use it here. So we have our same answers where we found out that Q equals 0.37 and that our P was 0.63. So using this information, let's start setting up our scenario this time. So again, we have red and yellow, same turtles, 34 being yellow. So it's important to understand for this equation, for this part B, we are trying to figure out what percent for the genotypes. We have 3 genotypes we need to determine. We have R, we have big R, and we have little. So first, let's start with our big R's. So we have big, and we know that this is representing our P squared. And P squared, P, which we just learned, was 0.63. So we're just going to square that number to find the answer for P squared. So for us today, that is going to be 0.3969, which is going to be 39.7%. So this number we just got is P squared. So that is the first part of our equation. So we still need to figure out 2PQ and Q squared. So we have our first genotype done. So the second genotype that we need to figure out is going to be big R, little r. And big R, little r is representing our 2PQ. As we just got on the last page, we know that we have answers for P and Q, so we're just going to fill it into the equation. So we have 2 times P, which is 0.63, times our Q, which is 0.37. So if we do the math for these, we're going to get that big R, little r is 0.4662, which is 46.6%. And big R, little r in our equation, our 46% is the 2PQ. So we have Q squared left. And in terms of our genotype, we need little r, and little r is our Q squared. So we already figured out Q from the last slide, and Q is going to be our 0.37 squared. If we do the math, this is going to give us a number of 0.1369, and so that is going to be 13.7%. And so we have finished off our equation. Our Q squared is our 13.7. So these numbers, our P squared, our 2PQ, and our Q squared, are representing the percentage of each genotype in the population. So we know that big R is 39% of the time, 46% of the time, and then our little r homozygous recessive is 13% of the population. The last 2 slides we're going to talk about here are just some strategies for helping you solve Hardy-Weinberg equilibrium equations. So if our population is in Hardy-Weinberg equilibrium and you are given the phenotypes, so we are getting so many are yellow, so many are red, that's the phenotypes, we are first going to want to find the frequency of our homozygous recessive genotype. So that's our Q. From here, we can take the square root of Q, and this is going to help us find P. So we can use this initial equation and find what our P is. Then we can plug that into our more complex equation to find the other genotype frequencies. And the second tip we have here is that if you're trying to figure out if a population is in Hardy-Weinberg equilibrium or not, and we're not sure, the first thing we want to do is use the actual numbers to calculate the genotype and the allele frequencies. We would want to look back at our practice problem number 2 for this. So once we have determined what P and Q are, then we can use our equation to find the genotype frequencies in the population. And we can use these two equations, and if the genotype frequencies don't match up with the actual frequencies initially calculated, then the population is not in Hardy-Weinberg equilibrium, and that means that this population is evolving. If they do match, then that means that you are in fact in Hardy-Weinberg equilibrium.